reaction, oxidative phosphorylation is very important and crucial phenomenon of life science, because at the end of this process, ATP is synthesized. So first of all, what is ATP? ATP is energy possessing molecule which is required whenever there is a need of energy or energy demands. ATP fulfill that energy demand. So ATP is synthesized as a result of transfer of electrons from NADH and FADH to like molecule to the oxygen via a series of electron transfer or electron carriers. So with the help of cellular respiration process, ATP is synthesized. So a cellular respiration process comprises of glycolysis, linked reaction, TCA cycle, which ends up with the oxidative phosphorylation. So at the end of glycolysis, linked reaction and TCA cycle, the net ATP or the currency of energy, that is ATP and GTP, are formed, but that are in low amount. So to fulfill, which cannot fulfill the demand of energy required by the cell or cellular mechanisms. So NADH and FADH2 like molecules which are high energy possessing electron transport or electron carriers. So they enters in the electron transfer flow or electron transfer chain system. So here what happens, NADH and FADH2 like molecules enters from complex number 1 and complex number 2 to the intermitochondrial membrane. So here this is the outer mitochondrial membrane, this is the intermitochondrial membrane, and this is the intermembrane space between the two layered membrane. So what happens here, NADH and FADH2 produced from the glycolysis, linked reaction, TCA cycle enters in the electron transfer chain system. What happens here, NADH converts to NAD plus via complex number 1, FADH2 converts into the catalyzed into the FAD plus via complex number 2, and at the end of this reaction, electrons are generated. Electron enters the electron transfer flow, and these electrons are accepted by ubiquinone, and this ubiquinone reduced to ubiquinol, and from ubiquinol these electrons are transported from complex number 3 to complex number 4 and finally accepted by the last acceptor of electron transfer chain, that is oxygen, and hence water is produced. But what happens here, electrons are energy containing entity, so this pumps the protons from matrix to the intermembrane space. So as a result, at the end of this electron transfer flow, concentration of protons increases in the intermembrane space, whereas concentration of proton decreases in the matrix. So a proton motive force is generated. So with the help of proton motive force, these protons tends to equilibrate themselves via transportation from intermembrane space to matrix, but they cannot do so via simpler diffusion process. So they need the help of ATP synthesis enzyme. So if we zoomed this structure here, so what this structure tells, it is comprises of F0 F1 particle. F0 particle comprises of A, B and C subunits, whereas F1 particles comprises of alpha, beta, gamma, delta and epsilon subunits. So it is also present in the intermitochondrial membrane, and this is the intermembrane space and this is the matrix. So with the help of A subunit of F0 particle, protons enters from intermembrane space to matrix, because here A subunit of F0 particle acts like a proton channel. So proton enters in this way, but here another important phenomenon also occurs. What happens here, when protons enters from this to this, via help of A subunit of F0 particle, A rotates the C ring of F0 particle, and then this C ring allows to rotate central stalk, that is gamma. Gamma acts like a central stalk between the F0 and F1 particle, which helps to combine them together. So once this gamma central stalk is rotates, it also allows to rotate the alpha and beta subunit of F1 particles. Now, it is a very well known that the ATP synthesis enzyme having the ATP synthesis property, but the actual catalytic activity is having with the beta subunit of F1 particle, because beta subunit exists in 3 different conformational states, that is loose, tight from intermembrane space to matrix, it allows to rotate the C-rings allows the rotation of gamma central stalk, and then from gamma central stalk allows the rotation of alpha beta subunits. So when beta subunit rotates, first of all what happens in the loose conformation state of beta subunit, ADP and inorganic phosphate are loosely bound with this beta subunit. Here they cannot form a bond between them. So once again, when another second proton pass from intermembrane space to matrix via a subunit of F0 particle, once again C ring rotates, then C ring rotates, allows the rotation of gamma, then gamma followed by the beta subunit rotation. Now beta subunit exists in its second conformation state, that is tight state. So here it is beta subunit tightly hold the ADP and phosphate together, and hence covalent bond is formed between ADP and phosphate, and ATP is synthesized. But whenever ATP is tightly bound to the beta subunit of F1 particle, ATP is of no use because ATP should be in free form. So another proton when enter from intermembrane space to matrix, once again C ring rotates, followed by gamma central stalk, and then followed by beta subunit rotation. So now it changes into the third conformation state, that is called open state. Here it no longer bound with the ATP, and hence ATP is released out, which can be used in the different cellular functions like metabolic pathways and cellular signaling. So at the end of this, we can conclude that ATP is synthesized at the end of cellular respiration, and whatever the ATP net ATP synthesized at the end of glycolysis and TCA cycle are in very low amount, which cannot fulfill the demand of cells or cellular process. So NADH and FADH2 indirectly helps in the synthesis of ATP by entering in this electron transfer flow system, and a proton motive force is generated. These protons tends to equilibrate themselves and pass from intermembrane space to matrix with the help of ATP synthase enzyme. And once protons enters from intermembrane space to matrix, it rotates the beta subunit conformation states. And so with the help of binding change mechanism of beta subunit, finally ATP is synthesized, and more amount of ATP can be produced. So from breakdown of one glucose molecules, total 32 ATPs are produced at a time, which can fulfill the demand of cellular processes. So this is all about the oxidative phosphorylation. Thank you.